Okay, so before I forget to say it, there were a number of important emails that you got this morning. The one about the mask mandate. In practice, you're still wearing them pretty much everywhere, except for, I don't know, maybe always you don't have to wear masks, but we'll see. Hopefully, you know, even less as we go forward. The other email that came through is there is a new drop date, March 25th. So if you are thinking about dropping, you have a little bit more time to make your decision on that. So enough time to get in another exam and see where you're at. So that's kind of nice. In addition, if you are thinking about dropping lecture, know that they will automatically drop you from lab because lecture is supposed to be a prerequisite for lab. So you have to get permission from your lab instructor if you want to stay in lab. It's just a, you know, it's just an email to our department assistant chair. It's not a big deal, but you need to get that override basically before you drop lecture. So just keep that in mind. You know, ask if you have questions. Also, a little advertisement for our BMS seminar this Friday. I know that's kind of a hard sell, the Friday before break. But this is an alum. Nico came through our department. Now he's at the University of Michigan. Apparently a great student, great guy. So it might be a good talk if you're around. We would encourage you to come just so we can have some more bodies here to support him. Right, and that's over in Mackinac at noon. Right. Okay, so last time we finished antimicrobial methods. So let me give you a practice question. A question like this is a little more difficult to ask on an exam. When I ask you to put something in order, it gets a little bit wordy when I try to do that. But take a look at that and see what order you would put those things in before we move on here. Okay, so hopefully we can agree on the likely least effect. Okay, cold water rinse of the skin. I know we've all done it. Oh, you throw your hands underwater. Oh yeah, I washed them. That really doesn't do a lot of good. You need that kind of degerming action, that surfactant action that we get from soap. So that's not terribly effective. Next, I would say probably hydrogen peroxide use on the skin. All right, so if it's used on the skin, that's an antiseptic. So that's probably 3% hydrogen peroxide. Okay, usually anything that's an antiseptic is just going to be, you know, kill your kind of more lower level microbes, and you know it has to because it has to be safe for the skin. I would say probably bleach would be 3. Okay, bleach, a disinfectant, but we considered it a kind of medium or mid-level antimicrobial, not sporicidal. Okay, so then hydrogen peroxide used as a vapor, and that as a vapor, I think it's usually like 35% hydrogen peroxide used as a vapor. And at higher concentrations, hydrogen peroxide was one of those high level antimicrobials. So that's considered sporicidal. All right, so, just kind of, there's some terminology there that you kind of get used to using correctly, hopefully. Talks about a lot of specific uses for those chemicals in particular. Okay, so where we left off last time, we just did a little introduction. I was a little rushed at the end there, but we talked about the death rates and how, and these were slides again that I didn't post, but how in 1900, one out of 3 kids died before their 5th birthday. So when you think about, if you hear somebody say, oh, the good old days, they were not thinking from a public health perspective. That sounds like frightening to me. But now, and COVID aside, our death rate, certainly from infectious disease, again, before 2020, has really plummeted. Big advances in hygiene, so especially treating our water. We get less infections because we treat our water. We're going to talk today about antibiotics and into next time, and then the use of vaccines. So really, again, COVID aside, and we have made tremendous progress in treating COVID. And I think that we'll get there, but we'll talk more about that. So pretty low level of infectious disease. But it will never totally go away. So we need to keep vigilant. Of course, there's problems with resistance to antibiotics. So there's still got a long ways to go, still more progress to make. All right, so as we get into chapter 10, we're going to start with a little bit of terminology, and then I always like to talk about things a little historical perspectives. So some people that have made advances in treating infection, in treating bacterial disease. Let's talk briefly about where antibiotics came from, what produces those, what kind of organisms, and we're going to think for a bit about what are some things that we have to consider when we decide basically whether antibiotics should be used or not. So what kind of concentrations do we use them at? How do we administer them? Are they going to be effective? So some things, all those things we want to think about their mechanisms of action. So we'll talk about some sort of in general, and then we're going to look at, I think I counted 14 different antibiotics, and think about especially their mechanism of action, different uses that they have, just some interesting things about them, side effects, etc. And then our discussion wouldn't be complete unless we talked about resistance to antibiotics. Before the pandemic, that was probably considered the #1 health emergency, health crisis that could come up. Worrying that we are going to run out of antibiotics that are effective because there's so much resistance. So we'll talk about that probably after break. We won't get to that till after break. All right, so we want to be square on a few terms. When you use the word chemotherapeutic drug, most of us think chemotherapy, we think cancer drugs. But used correctly, that refers to any drug that's used to treat disease. Okay. And then we can refine that further. If we say an antimicrobial agent, that's a drug that's used to treat some kind of infectious disease, whether it's viral in origin or fungal, some kind of mycoses, or whether it's bacterial. And then if we use the term antibiotic, okay, that refers to a drug that is produced by organisms, in other words, biosynthesized, and targets bacteria. Okay, so really targeting bacteria is kind of the really important couple words there. Because some of the antibiotics that we use now are not a natural product from some other organism. Sometimes we've kind of doctored part of their molecule, and we call them semi-synthetic antibiotic. So we've taken that natural product of some fungus, for example, and modified it a little bit in order to give that drug a little bit better properties. Maybe it's useful to work against different kinds of bacteria. Maybe it's more likely, maybe bacteria less likely to become resistant to it if we doctor it a little bit. There are some antibiotics that are totally synthetic. Okay, so there are a few drugs, only a few in use actually, that are just manufactured and they're not secreted by other organisms. We still call them antibiotics because they target bacteria only. Okay, so if a drug is an antiviral, we could call it an antimicrobial agent, but we wouldn't call it an antibiotic. Okay. And most of our drugs are either the natural product or semi-synthetic. Most of the ones in use right now. All right, so a little history lesson. Paul Ehrlich, we mentioned him before, back our very first lecture, I believe. I have trouble spelling that. Ehrlich, okay, so German scientist, he was a student in Robert Koch's lab. So you're thinking, you know, date's not important here, but late 1800s, early 1900s. We called him before the father of chemotherapy. So he was the first one, at least we give him credit for starting to really think about, hey, a patient has an infection, what can we give that patient to make that infection go away? And he tested many, many different compounds to try to find something that would work against the bacterium that caused syphilis. Syphilis is a particularly nasty sexually transmitted disease because it has neurological complications often and is more often deadly than some of the other sexually transmitted diseases. So it was a pretty bad one. He came upon a compound that he called salvarsan. It was a compound that had arsenic in it. So arsenic is actually really toxic to us. So it had kind of limited, it had some benefit against syphilis, but you know, again, it was pretty toxic to humans because it had arsenic in it, but it was kind of a good first step. We didn't really come up with any other agents that treated bacteria until the 1930s when Gerard Domack, he was another German scientist, and he was working at the chemical company Bayer and was trying out many, many different compounds to try to get one that would work against streptococcus. And he finally found one that he called Penecillin. Very effective, again, against really a specific kind of bacteria, streptococcus. But this was a miracle drug because up until that time we had nothing that worked. So things like scarlet fever was caused by a streptococcus. That was kind of a infection that caused high mortality, especially in kids. Winston Churchill took some Penecillin in the 1930s and credited it with saving his life. So, you know, these things certainly alter the course of history. Gerard Domack was awarded the Nobel Prize in Medicine, I think in 1929 and or 1939. Hitler wouldn't let him go accept it because he was mad at the Nobel Committee. So he never actually got the prize money, but he did get a Nobel Prize for that work. And then probably more famously, Alexander, I want to say Hamilton, Alexander Fleming. That's his very serious looking picture there over the side. he was working in his lab at a hospital in London. The story goes, he went on vacation and kind of threw some petri dishes off to the side. At that time, they had glass petri dishes, and so they had to be cleaned out. And he didn't bother to do that. He went away on vacation for a couple of weeks. And when he came back, he saw some plates, one plate in particular that looked something like this. Okay, chance favors the prepared mind. There was a mold growing on his plate of Staphylococcus, and he noticed there was a zone around that mold that was keeping the staph from growing. Okay, so he realized that that mold was secreting something, some important compound that could be antibacterial. So he isolated the compound that he thought was antibacterial. It's from a Penicillin mold, and so he called it Penicillin. But he couldn't get it, he couldn't make it in large enough quantities so that it would be able to, enough of it to treat an infection. So he wrote it up in a paper and then moved on and started to do research on something else. So that was in 1929. In about mid-30s, a couple scientists working at Oxford then, they saw World War II on the horizons. And of course, infection kills more soldiers on the battlefield than the actual blow wounds. So they realized they really needed to find something to treat bacterial infections in order to save lives. And so Howard Florey started searching the literature to find something, you know, anything that somebody might have found that might, you know, might be worth looking into further to develop something antibacterial. He came upon Fleming's paper and decided to assemble a team to see if they could mass produce penicillin. Okay, so Howard Florey and Ernst Chain, that's their pictures right here. Here's Florey and Chain. Chain was an organic chemist. So it's basically an organic synthesis to kind of grow up that mold in large enough amounts and then extract that active agent. And it was a hard, difficult process. They finally figured it out. And with the help of lots of other people, they were able to produce penicillin in large enough amounts and, you know, make it available to everybody, not just soldiers during World War II. Okay, so lots of people worked on that, but you can only have 3 people get the Nobel Prize. So it was Fleming, Florey, and Chain that ended up with the Nobel Prize. Okay, so that's just kind of where we got to where we are now. And then, you know, after penicillin, lots and lots of other antibiotics were found and developed, obviously. What kind of organisms naturally secrete antibiotics? Bacteria and fungi. Okay, not algae, not protozoa. It's all we found are bacteria and fungi, and certain species. We don't really understand why they secrete antibiotics. You would think, well, then they can kind of outcompete other organisms around them, you know, have better access to nutrients. But in nature, we don't think they secrete the antibiotics in high enough amounts to really be able to kill other organisms. So our best guess is that it's more of maybe a signaling function. But we don't really know. You would think if they're going to make the effort to make it and invest the energy to make it, that it would be for a good reason, but we're not really sure. But it certainly has benefited us. A couple genera of bacteria that have given us a lot of medically useful antibiotics, Bacillus, and these are all organisms that live in the soil. Okay, that's the one thing that they have in common. Okay, so Bacillus, a lot of you, I had some of my students just swipe places in the room and see what grows up on a petri dish, and then we gram stain those. And almost every student that swiped the bottom of their shoe found some Bacillus. Okay, so very prevalent in soil. Streptomyces is a really interesting genus of bacteria. So here's a light microscopy photo of Streptomyces. They grow in these long filaments, and they actually produce spores that are reproductive. Okay, so they act more like a fungus, but they're prokaryotic cells. So they're considered bacteria. Okay, and probably half of the antibiotics that are in use now are secreted by some species of Streptomyces. So really common producer. And then of course, the fungi, the one that we've all heard of now, Penicillium mold, they often have kind of a greenish tinge to them. The original Penicillium mold that they used to make original penicillin came from a rotting cantaloupe. Okay, so you can find these in lots of places in nature. Acremonium, that's what this one is down here. Not as probably familiar to you, cephalosporium. It used to be called cephalosporium, and we have a whole group of antibiotics called cephalosporin antibiotics, but they changed the genus name there to Acremonium for some reason. So it's just a few examples, again, of organisms that secrete antibiotics. All right, so what kind of things do we need to be thinking about? What makes a good antibiotic? How do we prescribe them? The number one thing that we have to keep in mind when we're using antibiotics, or again, trying to figure out whether they should be made, is that antibiotic selectively toxic? In other words, is it going to target some kind of pathway or structure that we as hosts are going to lack? Okay, if the antibiotic hurts our cells the same as bacteria cells, then that's not selectively toxic. Okay, we want the so-called magic bullet. We want it to target our enemies and not our friends. So we want to target that bacteria. We want that antibiotic to bind to some structure or stop some pathway that we don't have. Okay, so for example, I'll come back to this slide, but your book has a nice figure here. Mechanisms of action of antimicrobial drugs, or well, it calls it microbial drugs. Okay, we're going to focus on the bacteria, so don't worry about this one because that's a viral mechanism of action. But thinking, okay, why is this drug selectively toxic? The most common mechanism of action here is that antibiotic inhibits cell wall synthesis. Okay, they are almost always going to hinder something about peptidoglycan synthesis. Okay, so remember why that's selectively toxic. Animal cells, like us, human cells, we don't have cell walls at all. And even, plants or fungi that have cell walls, they don't have peptidoglycan in it. So we are targeting bacteria only here. A lot of our antibiotics are cell wall inhibitors because, again, they're generally pretty safe for us to take. Probably the second biggest class are drugs that inhibit protein synthesis. And don't worry about the ones that they list here because some of those we're going to talk about and some of them we're not. Don't worry about the ones that we're not going to talk about. Remember, bacterial ribosomes have a slightly different structure than our ribosomes. Okay, bacterial ribosomes 70S, we have 80S ribosomes. So remember, at the ribosome, that's where proteins are made. So we've got the green, proteins being made, and the red messenger RNA going through the ribosome. All right. So the drugs that most of them target actually the small subunit of the ribosome and bind there in order to stop protein synthesis in bacteria. A much smaller group are drugs that will disrupt cytoplasmic membrane. Membranes of bacteria are very similar to our membranes. So we don't have good selective toxicity there. There are a few drugs that we just use topically and would only be ingested if the patient was going to die without it. So usually those are just used topically. And then there are a few drugs that will inhibit some metabolic pathway that bacteria have that we don't. Okay, and then the last mechanism is either inhibit DNA synthesis or RNA synthesis. Some of those enzymes that bacteria use are just different enough from ours that we can get some selective binding there. And that's kind of the other most popular mechanism. Okay, so those are ways, again, we want good selective toxicity. Now, with any drug that you take, though, even though, say we don't have the structure that's being targeted, there are potential side effects, right? So a patient could develop an allergy. So an allergy is an inappropriate immune response. Okay, so, this is only going to be certain people that develop an allergy. As we get into talking about our immune system, hopefully it will become a little bit more clear how there's just genetic differences in one person's immune system from another. Differences in what kind of molecules they're going to recognize. So, differences among people as to who has an allergy and who doesn't. Some drugs are going to have a little toxicity. So kidney toxicity is a common one. A common concern, and we have to make decisions about, well, is this considered safe or is it not considered safe? And then disruption of normal microbiota. So remember, we have lots of bacteria in our body that are not harming us, but probably helping us, contributing to our health. So when you take antibiotics, it can cut back on the variety maybe of the bacteria that are normally there. So that can potentially cause problems. It can cause things like a patient, maybe if they have some Clostridium difficile in their large intestine, now that C. diff can overgrow. And you get this really nasty diarrhea as a result of taking antibiotics. You can have an overgrowth of fungi, candida fungi in the mouth, and that causes a condition known as thrush. So right there, kind of a whitish growth on the tongue and roof of your mouth. One of my colleagues gets thrush every time she takes antibiotics. I think she just maybe normally has a little higher amount of fungi in her mouth, and it just tends to overgrow every time she takes antibiotics. Same thing, a vaginal yeast infection. I didn't give myself very much room to write there. But again, when our normal microbiota gets disrupted, those are just a few examples of kind of side effects of that. We like to think about the spectrum of activity of the drug. So meaning, what kinds of organisms is it likely to kill? And do we consider it a narrow spectrum or broad spectrum? There's no hard and fast kind of definition there, but a narrow spectrum drug would kill kind of like a very small amount of organisms, or maybe just a certain species, ideally. A broad spectrum drug would kill lots of different kinds of bacteria, maybe gram-positive and gram-negative. So there are disadvantages and advantages to both. A broad spectrum antibiotic would be a good choice maybe for a wound infection when you have no idea what was causing it. You just need to get a drug to kind of get rid of whatever it is. Disadvantages, they're more likely to have, one of these, like get C. diff or thrush or something like that after taking out, some of your good bacteria as well. A narrow spectrum drug is better to use if you know exactly what's causing the infection. And then you kind of selectively take out that one organism and don't worry about harming your normal microbiota. Are the organisms that you're trying to kill susceptible? to that. Okay, and there's a way that we figured that out. There's a test we do. We just did it in lab this week. What's the name of the test? Kirby Bauer, thank you. Okay, so we can see if organisms are susceptible by doing a Kirby Bauer test. All right, so if you're not in lab, this is what a Kirby Bauer test looks like. You make whatever organism it is that you're trying to test. Maybe you take that from a patient that has some infection that you can't get a drug to work for. And you make a zone of lawn of bacteria on your media, and then you put your antibiotic discs on there. Here we're testing like 6 or 7 different antibiotic discs. You incubate that, and then you measure the zone of inhibition. So you measure one side to the other, okay? And then you compare that to a chart to see if that's big enough to be considered for the organism to be considered susceptible to that drug. There's differences in diffusion rates. So what's considered susceptible is a little bit different for different organisms, different drugs. But in general, see, there's a few discs here where you don't have any zone at all. Okay, that drug's not having any effect on that organism, so they're going to be resistant. Okay, and then generally, the bigger the zone, generally the more susceptible. Okay, a few other things to keep in mind. What concentration of the drug are you going to administer to the patient? Okay, and we have to keep in mind, we want to give it a large enough concentration to kill the organism, but not to have toxic effects on the host. So we like to understand what's called the therapeutic index of the drug. Okay, and how you come to that number, you take the maximum dose of drug that a patient can tolerate. Okay, so without having unacceptable side effects. And then you compare that to the lowest dose that will inhibit the pathogen that you're trying to kill. Okay, so the dose that a patient can tolerate versus the dose that you need for it to work against the organism. So ideally, you want this ratio to be as high as possible. So say a patient can tolerate 100 milligrams and it only takes 1 milligram to kill the bacteria, then your therapeutic index is 100, and that's considered very, very safe. Okay, as that window shrinks, say you needed, 10 milligrams, whatever, to kill the pathogen, then your therapeutic index is only 10. That's really not considered terribly safe. And I wish I've searched for lists of therapeutic indexes of different drugs, and I've had trouble finding them. I know morphine is considered very safe, and it has a therapeutic index of 70. But again, as that therapeutic dose gets closer and closer to the toxic dose, then that's when we have to be really careful how we administer the drug. And that toxic dose doesn't necessarily mean what kills a patient. It just whenever side effects are unacceptable. So if you're giving a drug for cancer and it makes a patient's hair fall out, well, you're trying to save their life, so be it. If you're giving a drug for strep throat that makes a patient's hair fall out, then that is not considered an acceptable side effect. So that can be kind of a moving target. Okay, so that relates to how we administer drugs. Okay, so the different ways that we administer antibiotics, this figure 1013 shows that. Drugs that we take at home, we would most often take orally, right? So that just means you swallow them. Also, we sometimes give antibiotics by shot or intramuscularly. And we also give them by IV, continuous intravenous. Okay, the third way or the fourth way that they don't mention here is topically. So sometimes we just put antibiotics on our skin for skin infection. So comparing those different administration methods, and if you look at the relative concentration of drug in the blood, that's the y-axis, you can see when we swallow pills, it doesn't really get the concentration up really high. Sometimes that's fine, but sometimes it's hard to get it like exactly where it needs to be. If we give it by shot, we can get the concentration up a little bit higher, but it tends to dissipate fairly rapidly. If we give drugs by IV, we can dial in that concentration to whatever we want, and we can sustain it for long periods of time. Okay, so when we're thinking about which way we should administer a drug, we have to think about the therapeutic index, because if it's a drug with a low therapeutic index, it's not very safe. You are not going to send the pills home with a patient and say, give directions on how to take them. Because if they don't follow directions and say, take 2 pills instead of 1, we don't want it to kill them. All right, so a drug that has a low therapeutic index, you're not going to give orally. You're only going to give that probably by IV or maybe by injection. You have to think about, is the drug going to be susceptible to stomach acid? Okay, this, you know, if the drug is altered when it passes through our stomach, then some drugs that just makes them not effective. So then if what we call that, if it's acid labile meaning altered by stomach acid, then we can't give it orally. Okay, so it would have to be more likely either IV or by shot or topical. And the third thing to think about is that some drugs just are not absorbed well. So they're not absorbed through the small intestine. Okay, so this was an early problem with penicillin, and it's why we couldn't give that first variety of penicillin orally, because it just didn't get to the tissues where it needed to go because it wasn't absorbed when we swallowed it through the small intestine very well. Okay, so have to think about all those 3 things. You know, it's a little bit more difficult to have a drug that you can swallow that's easy to take at home. And then resistance, of course, makes everything more complicated. There are some bacteria that either have intrinsic or acquired resistance. So if say a bacteria that doesn't have a cell wall, there are some bacteria without a cell wall. They are intrinsically resistant to a cell wall inhibitor. Okay? It's not going to do any good. There are a lot of drugs that work better on gram-positives because they have more peptidoglycan. And the gram-negatives, right, have this outer membrane that's just not as permeable. So we have less choices on drugs to treat gram-negatives. They have some intrinsic resistance. In lab, when we tested Pseudomonas, all those 4 antibiotics that we used, the organism was growing right up to all those antibiotics in Kirby Bauer. Pseudomonas just has some intrinsic resistance. Bacteria also can acquire resistance, and we're going to talk more about this. They can often acquire it maybe by gaining a plasmid through horizontal gene transfer and acquiring the ability to maybe make some enzyme that can make the antibiotic not effective anymore. So we'll talk more about that. And production, ideally, it's going to be inexpensive, because if a drug is super, super expensive, it's just not going to be commonly used. So. Okay. All right, so our first big class of drugs that I want to talk about are ones that inhibit the cell wall synthesis, and there's a huge class of drugs that we refer to as the Betalactam antibiotics. okay, or just beta-lactams. So you've probably heard of those before. These are a huge class of drugs that when you look at their structure, they have one thing in common, and that they have a beta-lactam ring. So this little shaded in green ring here, that structure, that's called a beta-lactam ring. Okay, so any drug that's considered a penicillin is going to have it. There's a class of drugs called carbapenems. They have it. Cephalosporins. There's some drugs called monobactams. Okay, those are all beta-lactams, and they all work by the exact same mechanism. Okay, and what is that mechanism? Here's your book's figure here. They inhibit peptidoglycan formation. So if you think back about a month or so when we talked about cell wall structure, we talked about there are enzymes. So when the cell is growing, the cell wall has to expand when the cell is growing. It has to do that really carefully. It has to make cuts between those NAM and NAG sugar subunits of peptidoglycan, and then it inserts the new sugar subunits, and then it needs other enzymes to come in and seal up those bonds. So it was autolysins that make those cuts between the sugars, and then if you remember transpeptidases, those were the enzymes that cross-link those NAM subunits with the amino acid crosslinks. Okay, so the top here, this is cell wall growth without any exposure to beta-lactam. So you've got the cutting, the inserting of the new subunits. Here it's kind of hard to see this, but they've got outlined in white these new subunits that have been synthesized inside the cell, brought through the cell membrane and inserted and made the cell wall bigger. Okay, so beta-lactams are only going to be effective when the cell's actively growing. Okay, if down here, these little green dots here, if that bacteria cell is in the presence of some beta-lactam antibiotic, these big green spots are showing where those crosslinks should be made, and they're not, because the beta-lactams are actually binding to that enzyme and keeping those cross linkages from forming. So in effect, you get a peptidoglycan structure that looks like this. It's kind of wonky. It's not totally intact. And then when the cell is in some hypotonic environment, and water is coming into the cell by osmosis, that cell wall is not intact. And so it leaves the cell very susceptible to lysing due to that osmotic turgor pressure. Okay, so it's not really the antibiotic kills it, it's other kind of environmental pressures that are going to bring about the death of the cell. Okay, so if you kind of think about what's going on a more molecular level, this on the left, that D-ala. So amino acids have kind of two isomers, a D and an L. The D isomer is not common anywhere except in bacterial cell walls, but that's what the structure of two of those amino acids that make up those amino acid cross bridges looks like. And here's the structure of penicillin. Okay, so then if we superimpose those two, look how they exactly fit on top of one another. Okay, here's that beta-lactam ring nestling right up in here. Okay, so in effect, that penicillin is acting as a substrate instead of the D-ala and fitting right into the active site of that transpeptidase. So this is basically competitive inhibition, but we don't call it that because it's permanent. Okay, so it binds permanently to those transpeptidases and keeps them from making these cross bridges. Okay. All right, so as we move forward, we're going to start with some Betalactam. I'm going to run through a number of drugs. I think there's 14 of them that I want to talk about specifically. Some of the slides, it kind of looks like this. I have a lot of information in there, and I do, but I want you to focus particularly on how the drug works. So if you only memorize one thing, there's some memorization involved here. That mechanism of action. Think about advantages of using that drug, like what do we use it for? I try to give you an idea of that. Some of the drugs have serious disadvantages. So in particular, some side effects that we don't want. I'm going to talk about other things like what's the spectrum of action. I like to talk about that, whether it's narrow or broad, just to get you used to that terminology. So not that you necessarily have to memorize that for every drug, or how we will administer it. Again, I just want you to get kind of familiar with that terminology, and you don't necessarily have to know, oh, this has to be injected or this one has to be given topically or whatever. Okay, so, and I've also, I'll get to it today, but a way that I think is helpful to kind of organize all this information, I'll get to that as well. I mean, flashcards are good too sometimes, but I'm going to make you a concept map, and I think that will be helpful. Okay, so if we start with our narrow spectrum penicillin, Penicillin G, this is a drug that's rarely used anymore, but this was the very first one that Florey and Chain were able to mass produce. And what's the letter for? Well, they had different kind of products that they were different products of the mold that they were able to synthesize, and G was the one that worked the best. So that's, we have Penicillin G. Okay, so I like to give you structures of drugs. I don't have the structure memorized, but you should be able to look at this and say, oh yeah, here's that beta-lactam ring, so I know this is some beta-lactam antibiotic. I think that's kind of reasonable. You can look for some things like that. Okay, so this was, again, that original product of it was Penicillium chrysogenum, I believe, was the genus and species. Again, this was a miracle drug. We had very little other than sulfa drugs that would help with infections. It had its limitations, though. It was only effective against gram-positive infections. So it worked on some things that sulfa drug didn't, but still there was a lot of infections that it didn't help with. It was really cheap. Florey and Chain actually argued about whether they should patent this or not, and they decided not to, that would never happen today, right? You know, pharmaceutical companies have to recover their costs that they put into it, but right from the outset, it was cheap and it was available to everyone. When you administer it the right way, then it reaches, you know, the concentration that's necessary in the tissues right away. And there were very few side effects associated with it. Well, we'll talk about allergies a little bit in a minute. Some people had allergies, but other than that, not many side effects. Disadvantages. So Alexander Fleming recognized really within a couple of years of these being made that we were going to have problems with resistance. And a lot of people said, no, that's not going to be a problem. Well, obviously, we don't hardly use penicillin G at all because most organisms are resistant to it. And that's because this ability to make a particular enzyme has gotten passed around, right? So I'm going to look more at that on the next slide, what beta-lactamase is. So I'll come back to that in a second. That original penicillin drug also required injection because it was acid labile. Remember, labile means easily altered. Okay, so it was altered by stomach acid, so it had to be injected. And anyway, it wasn't absorbed very well through the small intestine. So for both of those reasons, it had to be injected. I think it would be really interesting over break that if some of you see your families to ask the question, what do you remember antibiotics being like when you were a kid? Because my dad, who was born in 1943, remembers the doctor coming to his house when he was sick and he had to get an injection of penicillin in his rear end, and it must have been a little bit traumatic. But that was his early experiences with penicillin. Me, a generation later, I remember taking penicillin as a liquid. It was like the worst tasting thing that you could imagine. Like, it made you gag. It was terrible. Where most of you probably remember taking antibiotics as a child and it tasted like bubble gum, right? So it's gotten easier and easier, right? So, you know, ask your parents and grandparents, you know, what they remember about that. The second, they had different, you know, varieties of penicillin. They found that if they grew that original mold on a slightly different medium, it made a little altered product. So penicillin V, that can be taken orally. So I know, again, I used to say, oh, they don't use that anymore either, but then my son was given it before he had his wisdom teeth out. So they gave him a couple penicillin pills to take prophylactically, just so he didn't get an infection before mouth surgery. So that has some limited use today, and it can be taken orally. Okay, so let me go back to this Betalactamase production. What are Betalactamase? It's easier. It's kind of easy to get kind of goofed up. Wait, what's making what here when you think about resistance? Okay, so here's some penicillin drug. And Betalactamase, sometimes we call these enzymes penicillinase. I usually refer to them as Betalactamases. These are, again, produced by bacteria. If they have the ability to do that. Okay, so the Betalactamase enzyme, how does that act? That acts on the penicillin, and it cleaves that beta-lactam ring. So what you see here is that beta-lactam ring is opened out just a little bit. And as a result, that penicillin doesn't fit into the transpeptidase active site anymore. Okay, so now the transpeptidases are going to be functional, and the bacteria survive. That's antibiotic exposure. Okay, so some bacteria acquire the ability to make beta-lactamase, which acts on the drug to basically alter the drug so that it's not effective anymore. Okay, so sometimes they gain this, you know, through some kind of conjugation plasmid. Okay, and they can pass it around. Okay, and we have several professors over in chemistry that that's what they do is research on different beta-lactamases here at Grand Valley. All right, so we'll be talking about that more pretty soon. I also want to talk a little bit about Methicillin. This is a drug that's not used anymore, and I try to keep my discussion to ones that are commonly used now. But this has some historical importance. This came out in 1950s. Considered, maybe we could consider it a second generation penicillin. And you can see it has that beta-lactam ring, and it's a little bit, structure is very similar, but we altered it a little bit. It's a semi-synthetic, so that this ring is at a little different angle. And originally we thought, well, this is going to block the action of beta-lactamases produced by bacteria. So it's going to kind of protect that beta-lactam ring a little bit. And it was in use for about, you know, at least 10 years or so. It had some kind of nasty side effects. About a third of patients experienced kidney inflammation. So it had some more serious side effects than the original penicillin did. And it was acid label, so it had to be injected. But the main reason that I want to talk about this is where we get the name of MRSA. So you've probably heard of MRSA. It's a strain of Staph aureus that's resistant to methicillin. And you probably think, well, what's the difference? We don't use methicillin anymore. But this showed up in about 1961. Again, this strain of Staph aureus that was resistant to methicillin. Well, so that's why we named it that. It's not just resistant to methicillin, though. It's resistant to pretty much all the Betalactam antibiotics, as well as other antibiotics that are commonly used. So although it's not, the staph is not any more aggressive than other strains of Staph aureus, but it's just hard to kill. So incidence is not quite as high as it was maybe 10 or 20 years ago, but it's still certainly a problem, especially when somebody gets a MRSA infection, after surgery, and it's not just on the skin. So that's where the name came from. And I want to pause for a minute and talk about allergies to penicillin. I've read different estimations on how many people that have penicillin allergies, anywhere from about 2 to 10% of us. And most of the time, that allergy doesn't seem very serious. It maybe show up the first time as a little rash, maybe get some hives. It goes away when you quit taking the penicillin. Now, sometimes it's more serious from that. Some of you may know Dr. Baxter. He teaches a different section of micro and lab. When he was a kid, he had hives for 9 months after he took penicillin. So if you've ever had hives, you don't want to have them. Like, it's not fun. So I can only imagine that would be pretty terrible, terribly itchy. And if you have maybe a little rash or some hives the first time you take penicillin, the second time you take it, you may be kind of sensitized now, and you could have an even worse reaction. So if you have some kind of reaction like that, if you got a fever from the antibiotic, that might not be easy to know if that was from the infection or the antibiotic. But you shouldn't ignore it. You should talk to your doctor about that, because again, the next time there's a certain segment of people that will produce IgE antibodies to that product that penicillin gets broken down to in your liver. And you can actually go into shock. So anaphylactic shock, that's when you have such an inflammatory reaction that your blood vessels get so leaky that fluid leaks out of them and your blood pressure goes way down, organs fail, people potentially die from that. So about 300 people a year in the US die from just taking penicillin. So, if you had, like for me, I had a little rash when I was about 5, and now, I can never again take antibiotics. Just, am I still, or I can never take penicillin. Am I still allergic? Maybe, maybe not, but no doctor wants to take the chance out of an abundance of caution. I take the $100 antibiotic rather than the one that's free at Meijer. So, but again, better to err on the side of caution there. Okay, and there are allergies to, any drug, any antibiotic. It's a little bit more common for penicillin, sulfa drugs. It's a little more common for those as well than the other ones for some reason. All right, so then we consider kind of our 3rd generation penicillins. We're able to be tweaked a little bit to be more broad spectrum. So amoxicillin was one of the first broad spectrum penicillins that worked not only on gram-positive infections, but also gram-negative. So gram-positive and gram-negative. Not every single bacteria, but a good number of gram-positives and gram-negatives. Most common use of amoxicillin is just for the kind of the run-of-the-mill childhood infections like ear infections, strep throat. Okay, so commonly given, that's the one that, you know, is given that's bubblegum flavor, that pink liquid. Can be used in pill form as well because it's acid stable. So again, either swallowed as a liquid or a pill. Absorbs well from the small intestine. Less frequent doses than some of the early penicillins that had to be given every few hours. So maybe like 3 times a day, I think is common. This has been in use since, I want to say the 1970s. So we've used amoxicillin for quite a long time. We do worry about resistance, and there is some resistance out there. So in an effort to prolong the use of amoxicillin, we have actually, usually it's sold in a form that's called augmentin. Okay, if you augment something, that means you add to it to make it better. Okay, so we've added something to amoxicillin to try to, again, make it better, prolong its use. And that something is clavulanic acid. This is a Betalactamase inhibitor. So my understanding is that it binds to the amoxicillin and kind of shields that beta-lactam ring so that enzymes, beta-lactamases, can't get to it to cleave it. So again, that's just extended the life, and most of the time it's sold in that form. All right, so there are a few other beta-lactams that I want to talk about, especially the cephalosporins. These are, if you look at the list of the 10 most commonly used antibiotics in the US, this one, cephalexin, makes the list. The cephalosporin antibiotics, they usually, there's a ton of them, and you can usually tell that they're cephalosporins because they have that prefix ceph. So ceftriaxone or cephalexin, for instance. And unfortunately, a lot of drugs will have their sort of chemical name, so cephalexin, but then pharmaceutical companies might tack on their trade name. Okay, so Keflex is the same thing as cephalexin. Keflex is the brand name that you might have heard of. Okay, so this is a beta-lactam, so it has the same mechanism of action of our other beta-lactams, binds to transpeptidases. The structure of it here, there's a couple of variable regions that we can monkey with. So that's why there's a whole bunch of different ones. There's actually our very early cephalosporins were the first generation. We're all the way up to 5th generation cephalosporins. So we've been, again, using these for a long time. In terms of what we use them for, it depends on sort of what generation you're thinking about. So the early generations had to be, the early ones could be taken orally, but they were very narrow spectrum. They only worked on gram positives. And as we have, again, monkeyed with them a little bit, they've become more broader spectrum, but a little bit more toxic. So we have to inject them. Okay, so we can kind of, again, dial in that dosage a little bit better. So the higher generations, like the 5th generation, I think 4th and 5th generation ones are also able to cross the blood-brain barrier. So it's not common for pathogens to be able to do that, but something like a meningitis can do that. So we need a drug to follow it there. Commonly cephalosporins are given to treat meningitis. And then the 5th generation is just kind of different enough from some of the other beta-lactams that it can be used to treat MRSA, usually effectively. And even some people with penicillin allergies apparently can take the later generation cephalosporins. They're just, again, different enough apparently that people can tolerate them. But again, most of the time, doctors are going to give you something else to just not chance it. But, okay. A few more cell wall inhibitors that are not beta-lactams and have a slightly different mechanism as to how they interrupt cell wall synthesis. Vancomycin is a drug that we've had for a while, since the 1950s. It's kind of famous to be an antibiotic of last resort because when we first started using this, we had a lot of issues with the purity of it, and there was a lot of side effects and kidney toxicity. We've gotten a little bit better at the organic synthesis there, and that's less of a problem now. But it is still a narrow spectrum drug, only works on gram positives. It's another one of the drugs that often is helpful in treating MRSA. It also is often used to treat C. diff. How does it work? You look at the structure here, and it's a huge structure. So it's actually just a physical block. Okay, it binds to those peptide bridges and keeps the transpeptidases from reforming those linkages. It doesn't get absorbed very well in our body, so it's usually administered by IV. The exception there is if we're trying to treat C. diff, we need to swallow it so it kind of gets to the large intestine where the C. diff are causing the problem. Okay, so I said it's often sort of a antibiotic of last resort. When we tried every other antibiotic and nothing else works, we try vancomycin. There have been strains showing up in hospitals that we call VRE. That stands for vancomycin resistant enterococci. Enterococcus is an organism that's very similar to streptococcus, typically not causing problems with healthy people, but we get all these hospital-acquired infections that are enterococcus. So if you have enterococci that are even resistant to vancomycin, that's considered a medical emergency. And we don't have much left to treat that. So it starts getting to be sort of an alphabet soup when you think about these different organisms that are highly resistant. Bacitracin is one that may look familiar if you're in lab. Again, another cell wall inhibitor, but those new NAM and NAG sugars, like I said, they're synthesized inside the bacterial cell and then are moved through particular channels through the cytoplasmic membrane. So the bacitracin actually blocks those channels so that those new subunits can't even make it out into the expanding cell wall. Also only targets gram-positive, high degree of kidney toxicity if this one is ingested, but it still has use as a topical. Okay, so it's one of those antibiotics that's in triple antibiotic cream. Okay, so we actually have 3 different antibiotics in there, and they all work by a different mechanism. So if you have a wound, usually triple antibiotic cream is a nice, it's a recommended way to keep your wound from becoming infected. As bacitracin, and then we'll talk about the 2 other antibiotics, again, that work by different mechanisms in there. Okay, don't be afraid to jump in if you have questions or comments, or a lot of you have certainly more clinical experience than I do, so don't be afraid to share a short story. Our last cell wall inhibitor, at least I would consider a cell wall inhibitor. We'll talk about that. Isoniazid. It's kind of fun to say, isoniazid. This is a fully synthetic drug, and it prevents the synthesis of mycolic acid. So mycolic acid is found in the cell wall, so I consider a cell wall inhibitor. You could consider it preventing a pathway that some bacteria have that we don't. It's very narrow spectrum because remember what kind of bacteria have mycolic acid in their cell wall? It's the mycobacterium. the acid-fast organisms, okay, that have that mycolic acid. So we always use isoniazid as kind of one of the first drugs that we try when we're treating tuberculosis or leprosy. So again, this is, you know, E. coli is going to be resistant to isoniazid because E. coli doesn't make mycolic acid. All right, so this is the beginnings of our concept now. So to me, it kind of has helped my brain kind of keep these organized. Here we have our cell wall inhibitors, and then some that are beta-lactams up here, others that are not beta-lactams but are still cell wall inhibitors. So we've got about 7 drugs there. Okay, so the next big class of antibiotics are the protein synthesis inhibitors here. So we've got several examples there. And then our 3 other mechanisms, nucleic acid inhibitors, cell membrane inhibitors, and metabolic pathway inhibitors. So we're actually about halfway through our drugs, even though that seems like we've only done kind of one mechanism there. Let's just start our protein synthesis inhibitors here. And I would encourage you to kind of fill in the drugs maybe as we go or later on as you go. You could fill in other things here like side effects or something like that, just to kind of help you keep things straight. In general, most of the protein synthesis inhibitor antibiotics are more broad spectrum. Okay, so they're working on both gram positives and gram negatives. That bacteria Streptomyces has produced a lot of different drugs in this class. So one of the big classes are the aminoglycosides. And the aminoglycosides, they bind to that small subunit of the ribosome, change the shape slightly there so that proteins aren't translated correctly. That's a little redundant there. I said broad spectrum again. They don't absorb well from the small intestine, and they are altered in stomach acid. So for that reason, we have to give them by injection. And we have toxicity problems, in particular with the kidneys and the ears. So I found this little mnemonic. It's kind of fun. The kidney is boxing with the ear, and written on the gloves are the different aminoglycoside antibiotics, or a few of them, like kanamycin. Most of the aminoglycosides end in this mycin suffix. Okay, so the kidney's boxing with the ear, and below it, it says common toxic effects, ototoxicity and nephrotoxicity. All right, toxicity with the ears and the kidneys. Okay, so again, they usually end in that myosin. We're going to look a little bit more closely at streptomycin, but I wanted to mention neomycin because this is our second antibiotic in that triple antibiotic cream. Okay, so we got a cell wall inhibitor and now a protein synthesis inhibitor, an aminoglycoside. Okay. All right, so let me just finish with streptomycin. This was the first drug that was effective against tuberculosis. So, you know, this infection that was the scourge of millennia, now we had something that was effective. So this was made soon after penicillin came out, just a couple of years after penicillin. The guy's name, his name was Selman Waxman, who was a soil scientist, and he actually developed about 20 different antibiotics. We now know that he took the credit for a lot of people in his lab that did a lot of work. But anyway, so here's the structure of streptomycin. So an aminoglycoside is an amino-modified glycosidic sugar. So sometimes we draw sugars in kind of this flattened form. So you can see the sugars, and then here's the amino groups, amino-modified glycosidic sugars. So when you see the structure, often it kind of makes sense by the name of it. Okay, so These bind here to the small subunit. The ribosome changes the shape a little bit. So then the wrong amino acids get incorporated. Okay, it doesn't read that messenger RNA correctly. Broad spectrum drugs, toxicity to the ears, that seems crazy. This will make sense to those of you in anatomy. It binds to the vestibular portion of cranial nerve number 8. Okay, so that can cause some mild hearing loss, maybe just some tinnitus or vertigo, maybe. It can cause permanent hearing loss and deafness. So depending on how much the patient is exposed to, we have to be very careful how we dose that out. And it's not used anymore unless the patient's potentially going to die because of that ear toxicity. So again, we dose it really carefully and we use it against really serious infections. It was the first drug that was effective against TB and bubonic plague. Yeah. Did he have tuberculosis or something?
I don't remember. I know he's my grandma. She said it was definitely he had some kind of infection, something like that. I assume it's something.
Let's have something serious. Yeah. Yeah. Okay. Good. Yeah. Thanks for sharing that. Tularemia is another really serious infection carried by the fleas of rabbits. Okay, so yeah, if you have comments, I'd be glad to hear them. Otherwise, I hope you have a great break, and I will try to be coherent on Monday. I hope you are too.